Hello, good day to you all, and welcome to today's section. This is PCR Molecular Diagnosis Program, um, PCR Molecular Diagnosis Training, and today we're going to be taking Module 1. And while we do that, I want to run through some things for us. This meeting is being streamed live. Okay. So just quickly run. So the goal of this training is to equip every participant with the skills, tools, and knowledge on PCR technique and molecular diagnosis with the current trend in the healthcare and the diagnostic industry. So at the end of the day, we should be able to equip with the skills, with the tools, and the knowledge on PCRO technique and molecular diagnosis. PCRO technique is one of those molecular diagnosis amidst others. Now, the objective is that at the end of the day, participants will be able to understand the concept of molecular testing and application in diagnosis. You should be able to understand the PCRO technique and its various application and platform that is currently in use. You should be able to understand the application of QMS in PCR molecular testing. Then through the practical section, participants should be able to apply the knowledge to any platform they encounter. Okay, so these are the modules we are going to be looking at. So introduction to molecular testing, that's module 1. So module 2, we're going to be looking at molecular technique. Module 3, we're going to be looking at sampling processing analysis for molecular analysis for different diseases. Module 4, we're going to be addressing quality control, quality assurance in molecular testing. And module 5, we're going to be looking at results and outcome of molecular testing. Then module 6, we're going to be looking at introduction to PCRO. Module 7, we're going to be looking at protein synthesis and genome. Then module 8, we're going to be looking at PCR and application in molecular diagnosis and monitoring. Then module 9, we're going to be looking at workflow and testing in PCR lab. Then module 10 will be addressing DNA and RNA extraction. And module 11, we're going to also be looking at QA and QC in PCR procedures. Okay, so these are the modules we're going to be looking at. We have different facilitators that are going to be looking at these modules all through this program. So please, you need to pay rapt attention to what we are going to be doing. And it's very, very important for us to know that this training does not only teach you on how to do molecular testing. There are a lot of aspects of molecular biology, molecular science that you might be interested as a business person, as an investor, as a career person, as an academic, or any other aspect as a researcher, do a lot of things. There are a lot. So don't just fix your mind on only testing. So that's why we call it PCR molecular diagnosis program, basically. So there are a lot of things you can be getting from this training. So please open your mind, open your thoughts, and learn as much as you can during the course of this training. So thank you. So today we're going to be looking at introduction to molecular testing. By way of introduction, my name is Dr. Aliu Israel. I'm one of the lead facilitator and the executive director of PcR Consultancy. This program is powered by PcR Consultancy, and we're bringing this training live to you wherever you are hearing us. So what is molecular testing? So as a professional, as a science-based person, when someone asks you what's molecular testing, what comes into your mind? I believe you have an idea of what is molecular testing, but generally, molecular testing or molecular diagnosis describe different testing procedures that detect a specific DNA or an RNA sequence. So molecular testing is beyond PCR. There are different testing procedures. PCR is just one. that detects specific DNA or an RNA what sequence. So in a simplest form, that is molecular testing. Now, when we talk about molecular diagnostics, what does it do? One, it helps in detection of genomic variants, aim to facilitate detection, diagnosis, and prognosis. So you can use molecular diagnosis to detect a uh, uh, particular disease, a particular DNA, a particular RNA. You can use it to diagnose it. You can use it to help in the prognosis, and you can also use it to monitor response to therapy. Someone is in a uh, particular treatment. With your molecular testing, you'll be able to know if the patient or the person is responding to treatment. Most importantly, it also helps for detection of genomic variants and genetic information. So a lot of molecular testing boils down to what detection of what of genomic properties, genomic informations, a whole lot of things, gene sequencing, a whole lot of things we want to look at. What are the genomic properties? What are the genomic properties of that uh, of that organism, of that department, of that what we're actually looking at for when it comes to what that testing. Now, what is the importance of molecular testing? Molecular testing has various importance. One, it helps to guide patient management from diagnosis to treatment. So you can use molecular testing from diagnosis to monitor to treatment and etc. It is vital in the field of cancer, like we have oncology, infectious diseases, congenital abnormalities, and etc. Let's say, for example, now, some when we want to detect congenital abnormalities, we have seen patients present with maybe two sex hormones, two different sex, and you want to know which of these sexes is dominant. You can use a what a molecular testing to be able to detect those things. A lot of congenital abnormalities can be detected through molecular testing. There are a lot of procedures in this molecular testing. We have PCR, we have fish analysis, we have a whole lot of them that we could use to detect this. So in Module 2, we're going to be looking at different molecular techniques there. So it helps in rising, due to the rising demand of genetic and genomic information, molecular testing is very, very important. You see that some person wants to know their DNA profile, want to know if they have the potential of even having a particular gene, maybe breast cancer, for example, or something like that. With molecular testing, you can detect such information even ahead of time. So it's due to the rising demand of genetic and genomic information, molecular testing is becoming relevant, and that helps in precision medicine. Rapid expansion of molecular technique within the clinical setting. So there have been a rapid expansion of molecular technique within the clinical setting, and this has given the great importance to what to molecular testing. Now, some of the progress in molecular diagnosis has been the rapid development and growth in molecular methods and in principles in this last decade. So there are lots of molecular methods that have been developed, lots of molecular method that is being used currently, and this development and this principle has been expanded in the last decades due to the pandemic, due to a whole lot of things, due to the current trend, due to precision medicine and etc. This progress has been made in molecular diagnostics. Now, the integration of new technology into the clinical molecular diagnostic laboratory has been critical toward advancement to the goal of achieving precision medicine. So one of the goal of precision medicine, what precision medicine has helped to achieve, is this integration of these molecular techniques. So we want to be precise about what we are doing. We want to not just not just looking at the proteins, but looking at the gene that has the characteristics of that organism, or for example, of that disease of etc. Let's say, for example, now, someone comes in to do a test like malaria test. You could do you could do a whole lot of tests like the rapid test for for the patient. You could like the rapid testing. You could do like the gold standard, which is the microscopy, which is the microscopy. You could go to do even do things like let's say the QBC, which is a concentration method, but you can also go ahead to do what PCRO, PCRO malaria. Now, it's possible for you not to be able to detect that malaria in the rapid if it does not have if the malaria is not up to some certain level. Now you could also mix it out during the microscopy, and even the QBC may not be able to pick it. But if that malaria is there, when you do a PCR molecular testing on them using PCR, you'll be able to detect it. Why? Because some of these techniques help to amplify the genetic material for you to be able to assess them. So it is precise. So that's part of precision medicine. Be able to achieve precision medicine. So we are not doing a guesswork. So if the malaria gene is there, is there. If the malaria is not there, it's not there. So the integration of new technology in the clinical molecular diagnostics laboratory has been critical towards advancement of the goal of achieving what precision medicine. Now, quality control and quality assurance is necessary for detection of problem and to monitor errors and to implement methods to assure quality. Every diagnosis, every testing, there must be quality control and quality assurance that is put in place to help from the pre-analytical to the post-analytical. So quality assurance are those programs, are the SOPs, are the processes, the EQAs, the training, everything you put in together, the system that helps to ensure that the result is accurate, the result is reliable, and the result is timely. That's key. Why QCs are those procedures? Those are programs that we do along as we are doing testing. Let's, for example, as you are doing extraction, there's a quality control program. As you are doing as you are doing application, as you are doing the testing, you are in the real-time PCR or whether it is gel electrophoresis you are using, for example, there are procedures, we call it quality control procedures, that you run side by side with the testing to ensure that what there is no problem and to ensure that the quality of results is guaranteed. Now, we have in the molecular testing, we have, just like your other testing, we have the pre-analytical, the analytical, and the post-analytical. That's the laboratory process. Now, in molecular testing, just like every other testing, we have what we call the pre-analytical consideration, like specimen collection. So there should be specimen should be well collected. People who are collecting specimen should be green. The specimen should be collected in the appropriate containers. The appropriate sample should be collected. Let's say, for example, if it's a blood sample that is needed, the blood should be collected. If it is sputum sample, it is sputum. If it is let's say, for example, it's a swab, that swab should be collected. So the appropriate sample collection. So these are very, very key, because when you don't have the right specimen and these specimens are not collected properly, a whole lot of misdiagnosis can happen. Now, transportation of the samples from, maybe, for example, because some of the time where these samples are collected may not be where they are analyzed. So the sample will need to be moved from one location to the other. Talk about storage. Sample being stored at the appropriate temperature, even the transporting in the right media. If you need a transport medium, you have to put it, how you package it, etc. We have like things like triple packaging and etc. So all these are considerations, and all these things we're going to look at when we were looking at module 3. Now we'll talk about storage. And the samples properly stored, how are they stored? They must be stored in accordance with the what operating procedures. They will also have a critical aspect of the pre-analytical consideration molecular test, which is what the nucleic acid extraction. Now, this nucleic acid extraction helps you to extract either the DNA or the RNA, depending on what you are actually focusing. So if you are extracting a DNA, you should have an RNA that is free from the DNA, or a DNA that is free from an RNA. All this we are going to discuss during the course of this training. Then we also have the clinical aspect, which is the analytical aspect. So there could be it could be a manual method, it could be an automated method of testing. It could also be both, depends on the downstream procedures and the facilities and a whole lot of things that you have at your disposal. So a lot of people still do some people still do manual method, some people still do automated method, depending on your downstream procedure. So their consideration, they must be standard operating procedures at this point, and staff must be trained adequately on how to do the testing, how to read the testing, how to ensure that the method are validated. But you don't just start running a test without validation. So all these aspects falls under the clinical aspect. So there must be a proper validation of the testing procedures. Let's say, for example, I have a PcR machine and I'll be running, let's say, HIV viral load there, and suddenly I want to start doing Lassa fever. Now, I don't just go and get a kit from the market. and run one or two controls and start. I have to validate. I have to validate that kit and validate that test method on that equipment, even though I'll be using it to run other tests. So all these things are part of what the clinical what laboratory, the analytical aspect. Then we also have the manual and automated method for nucleic acid extraction. Just as I've said, we have things like cell lysis, denaturation, and nucleoprotein complexes, inactivation of nucleases. All this we are going to have a better explanation and a good breakdown on all this in separation of nucleic acid from the remaining contaminants, example, protein and other nucleic acid, leaving the DNA-free RNA or RNA-free DNA. Let's say, for example, you are looking for the RNA, you are doing extraction, RNA extraction. So when you see a DNA, the DNA becomes what a contaminant. So all this separation of all the nucleic acid from the remaining contaminant are critical aspect of what the nucleic acid extraction. Now, when we have manual automated for different stages of the DNA extraction. So the DNA extraction and RNA extraction can also be a manual method, can also be what automated method. So we have the manual method, the automated method, application and product detection together with real-time PCR. Molecular laboratory will continue to become more efficient, cost-effective, and there are a lot of microarrays of technology, such as DNA chip, that will likely further increase the utility of the molecular detection in the clinical laboratory, whether microbiology laboratory or hematology laboratory. And one good thing about uh, molecular testing, it is one of the center of all testing. So whether you're in histology, it's applicable to you. Whether you are in chemical pathology, it's applicable to you. Hematology, wow, applicable to you. Microbiology, applicable to you. So we have it's a meeting point when it comes to the testing method for all of these various departments that we'll find among the laboratory. So what are the applications of molecular testing? One is for infectious diseases. You could use to detect infectious diseases. Test for genetic properties, for genetics. Molecular diagnosis is applicable in pharmacogenomics. Molecular diagnosis is applicable in oncology. It's applicable to a whole lot of areas. And it is the modern day testing method. Molecular is key. And that is why a lot of labs, a lot of procedures are driving towards precision medicine. You could see maybe like 20 years ago, let's say, for example, there are a lot of labs that, when it comes to molecular testing facilities, they don't have. But due to COVID and a whole lot of infectious diseases, drive for precision medicine, you see now a lot of facilities now have this as part of what their system, and making huge investment to ensure that they are able to what diagnose properly and have this test. Okay, so these are some of the applications of molecular testing. And hold on.